The last subject I want to discuss with you with respect to translation has to do with microRNAs. And as you may know, these are small non-coding RNAs that are found encoded in cellular or viral genomes. They're made by Pol2 or Pol3, and they're made as precursors, which are then processed to 21 nucleotide microRNAs. And our genome encodes over 1,000 of these microRNAs, and they have important roles in regulating protein synthesis. And the way they work is they bind the 3' untranslated region of messenger RNAs, and they either cause degradation of the mRNA or they repress translation. And this is where we want to talk a bit about this right now. These microRNAs function in a protein complex called the microRNP, and this, in addition to the microRNA, has a number of cellular proteins, one of them called dicer, which you may imagine can dice up the RNA to which the microRNA is pointing. And these microRNAs typically are complementary to the 3' untranslated region of the mRNA. And so here we have at the bottom the 3' UTR of mRNA, the end of the open reading frame. We have a poly A tail. And here's a typical binding site for a microRNA. It hybridizes in two regions with a bulge in between. It's quite close to the 3' end. And again, the amount of complementarity determines whether the mRNA is degraded or translation is repressed. The higher complementarity leads to degradation, the lesser leads to repression of translation. And we think that these affect translation in two ways. The mechanisms are still being worked out, but here we have an mRNA with these microRNPs bound to the 3' end. Remember, the microRNAs plus a couple of cell proteins. It's been found that the microRNPs bound to the 3' UTR can affect initiation. They can block cap recognition or even 60S subunit joining. Again, mechanisms to be determined. And they can also block elongation. So depending on the particular microRNA, can have an effect by causing ribosomes to slow down, for example, or even fall off. So that's how they regulate translation. I want to leave you with a couple of interesting examples of microRNAs and how they interfere or participate in viral replication. There's an interesting one in the liver called miR-122. It's liver specific, and it's absolutely required for hepatitis C virus replication. If you take it away, the virus can't replicate. And this is in part why the virus is hepatotropic. And right now, drugs are being developed to target this microRNA because it would be a good antiviral therapy. A number of microRNAs in the cell target either viral or cellular genes that are needed for viral replication. And these have been identified in various systems. You might be wondering what cellular genes would be needed for viral replication. Well, viruses, of course, have evolved to use different cellular genes in different stages of their replication cycles. And so there are microRNAs within us that regulate those genes and modulate virus infection. Now, on the other hand, viruses have evolved to take advantage of some of these miRs. For example, miR-141 targets the mRNA encoding EIF4E. So this is a miR that exists in the cellular genome probably to regulate EIF4E production. When enteroviruses infect cells, they induce the production of miR-141. And what does that do? That decreases the amount of EIF4E, and of course 4E is the cap-binding protein. So it shuts down cap-dependent, 5' end-dependent protein synthesis, which of course the enteroviruses don't care about because they have an iris and they don't need caps. This is brilliant. This is just brilliant. They take advantage of something that's already there and use it for their own good. Diabolical. Do viral genomes encode microRNAs? Some of them do. Herpes viruses have a lot of them, fewer in other viruses. And what they do is still being worked on. This is a very active area of investigation. We know already that Many of these block apoptosis. When viruses infect cells, the cells respond by killing themselves. And viruses want to block that because they want to replicate. Some of these viral mirs block apoptosis. They help the virus escape from immune responses. Here's an interesting one. Some of them prevent the arrest of the cell cycle. Upon infection, many cells stop dividing in response to infection, another antiviral response. Viruses, especially DNA viruses, want the cells to be dividing because they need the DNA replication machinery. So they have mirs to make the cells keep dividing. Amazing. And some of the mirs promote latency. That is, the ability of the virus to remain in the cell for long periods of time. So we have now taken these mRNAs, these central mRNAs, we've made proteins. Next time we're going to use those proteins to make particles. We're going to assemble virus particles, and that will complete our analysis of the viral replication cycle.